So I'll just tell you a little bit about myself. I'm a clinical geneticist, so my training is in, so I'm a doctor who's trained in clinical genetics as a subspecialty, and I've always been working in the area of intellectual disability. I have actually a child with a rare condition. It's not associated with intellectual disability, it's associated with rare cancer syndromes. But it took a long time for her to be diagnosed, even though I was a clinical geneticist. And I suppose that made me realise how my patients were going through this diagnostic journey of, taking a long time to get a diagnosis. And so I was sort of inspired to do this because I could see that my patients with their children were going through a similar journey to me, and it just sort of felt like something that I wanted to do. So I work in the New South Wales Genetics of Learning Disability Service. So this is one of my patients, and we have permission to show her photo. But this is Charlie and her mother Ainsley. And so Charlie has an undiagnosed intellectual disability. She's non-verbal. She's only just started to walk, but she has had exome sequencing and undiagnosed. And lots of our patients remain undiagnosed despite having exome sequencing, and I'll talk to you about that. But without, despite not having a treatment for a lot of these conditions, parents really are looking for a diagnosis because it can explain management, it can inform prognosis. Really, it's the first step towards targeted treatment in the future, and helps parents make decisions, obviously, about other children. And so we're still looking for a diagnosis for Charlie, and I'll come back and talk about Charlie a bit later. So intellectual disability. Yeah.
Can I just say I wish you were our geneticist?
Oh, thank you.
We were told that we were told that a diagnosis didn't matter.
I think the diagnosis really matters. Yeah.
100%.
Yeah, definitely. Yeah.
But I'll talk to you later.
No, that's okay. So intellectual disability is common. So 2 to 3% of children will have a form of intellectual disability or cognitive impairment. Probably moderate is closer to 1 in 200. And the most common cause is genetic. So it can be prenatal, environmental, but usually it's genetic. That doesn't mean it's inherited. So the gene change might have come from one or both parents, but often has occurred for the first time in the egg or sperm out of the blue by chance. And so the parents are unaffected, but they have a child who clearly has something that's going on that's different to the parents. Because they're genetics, there are thousands of different genes involved in normal cognition, and these conditions are incredibly difficult to diagnose. I say that they're collectively common, but although they're individually rare, they're collectively common. So there are as many people in Australia with rare diseases as there are with diabetes, but it's always been difficult because they're individually rare. So many of them have a prevalence of less than 1 in 2,000, and some of them, like you said, there might only be 100 families in the world. Sometimes there's only 2 families in the world. So 50% of these children will have facial features that can provide a clue to diagnosis, and this little girl here has a condition called Rubinstein-Taybi. So every child is uniquely beautiful in their own way. It's just that some of these children, when you put them together, you realise that they look similar. And so that's how we come across the word syndrome, which simply means a collection of features that occur together in a recognisable pattern. So this just is not a very good slide, but just shows a little bit about how we have all our genes inside every cell, and then we have our DNA, and that's a recipe that makes proteins. And we have proteins in our body for all functions in our brain, how we work, how we digest our food. But all these proteins, there's lots of thousands of proteins involved in normal cognition, and these are all coded by different genes. And really, this is the black box. We still don't know the majority of genes involved in normal cognition. So although we don't have a treatment for these conditions, understanding all these individual rare diseases helps us to understand the pathways, because there's going to be overlapping pathways, and will eventually help us understand towards targeted treatments, because treatments will likely be similar for some of these conditions that are within the same pathway. So when I started genetics over 20 years ago, the genome wasn't mapped. And so geneticists realised that children, some children look similar. And so they started clustering children based on their faces. And these conditions were initially named after the doctors, so Coffin and Dr. Lowry and Dr. Williams and Dr. Cornelia DeLang. And so these conditions were described. And when you look back at these old historical photos, usually the most severe children are the ones that were first described. And then what would happen, or most severely affected, and then what would happen is a few more doctors would go, oh, actually, I saw that in that article. Yes, I've got a patient who has Cornelia-de-Lange. And so we started then to build up this understanding of these conditions. And in those days, we didn't have any genetic testing. And so parents would come in, and if we could say, look, well, your child has Kabuki syndrome, then we could say, well, look, yes, that's not likely to happen again. Or if your child's got Coffin-Lowry, that's on the X chromosome, there's a 1 in 2 chance that another male will be affected. So they wanted this information to help with family planning. So in 2003, the genome was mapped, which really was the first revolutionized genetic testing. It was the first thing, because the next thing has just happened. So what we then found was all of our genome in every cell of our body had 2.2 billion letters of DNA, or these nucleotides. And so these are letters of DNA code, and they're in every cell of our body. But what we then found out, if all of our genetic material was like an apple, only 1% of that apple was actually coding for proteins. And so the genes, our 20,000 genes, make up this 1% of our genome, and that's what's coding for apples, for the proteins. So this is important because it helps you understand why it's very difficult to make a diagnosis. So we know we have 20,000 genes, but a banana and a mouse also has around the same number of genes. So what makes us different is not the number of genes, but the rest of the genome, which we call non-coding DNA. The other thing that's important is, even though we know we have 70,000 genes, we don't know the function of 70% of these genes. So we know they're mapped, but we don't know what they do. And a large number of those genes will turn out to be intellectual disability genes, but we just don't know what they do. So the rest of the genome is what we call non-coding. So probably 10 years ago, it was thought to be junk, but now we know the main difference between us and a mouse and a banana is not the number of genes, but the amount of non-coding DNA we have. And this is thought to regulate the 20,000 genes. It's a black box. So a lot of the patients who remain undiagnosed may potentially have either a change in one of the genes that we don't know what they do, or a change in this non-coding DNA. So eventually, after the genome was mapped, doctors were able to cluster, get groups of individuals together, and then they could compare their DNA, and they found that these conditions were due to single genes. And sometimes they were due to more than one gene. But we then started realizing that these single gene conditions actually had a gene that was responsible for them, which was quite exciting, because it meant that if we looked at a child and made a clinical diagnosis, we could then go, I've got a genetic test which will confirm what I'm thinking clinically. So this slide, is that the whole slide coming through there? Yeah.
Yeah.
So this slide shows an example of some children with some different conditions. So if these children came into clinic, say, 3 years ago, I would have to look at their face, and I would have to think, well, okay, I think you've got Kabuki syndrome. I'm going to test the gene for Kabuki syndrome, or I think you've got Pitt-Hopkins syndrome or Coffin-Lowry, and I'm going to test that gene. But testing was so expensive that it cost $2,000 per test, which meant as a doctor, I had to try and be pretty careful how I spent the health budget money, and I could really only order one test, maybe two at the most. And our diagnostic rate around the world was about 15%. So even though the genome had been mapped, we still weren't making lots of diagnoses. So the second thing that's really changed how we manage patients is the introduction of massively parallel sequencing. So this has really only been in the clinic for about 2 years, and probably it's now been a routine test for less than 12 months. So this is how new this test is. And what this test does, it allows us to sequence all of those 20,000 genes, which we call the exome, for $1,000, and the whole genome, which is like the whole apple, for $3,000. So that is amazing. So now, instead of me having to work out which one gene it is, I can test all the known, and there are 1,500 known intellectual disability genes in a single test. Has anyone lost me, or are you right? You okay?
No, it's good.
That's good.
Okay. So that was really exciting. And as a geneticist, I thought, well, I'm going to get a diagnosis for all my undiagnosed patients now, because I would bring them all back in and we do this new test. But what we found, that despite testing the 1,500 known intellectual disability genes, we're still only making a diagnosis in about 30%. Now, some labs will say it's up to 50, but they're very select patients that are, have lots and lots of problems. But for children who have moderate intellectual disability, who are coming in, who might have some mild facial features, the diagnostic rate is around 30%. So there's still a lot we don't know. And this 70%, or this sort of missing, they call it the missing genes, we don't know if they're part of those 20,000 genes that we don't know the function of, or are they part of that non-coding regulatory DNA. It's estimated that there are another 2,000 intellectual disability genes to be discovered. So the reason why so many people still remain negative is we haven't really characterised all these genes. Well, that's one of the reasons. So this test is now called whole exome sequencing, and it's now the first line investigation. So when I see a child now who comes into clinic with moderate intellectual disability, this is the test I order. And it will soon have a Medicare rebate, which will allow paediatricians to order this as a first line test for children with intellectual disability. So I shouldn't say that, it's getting there. So it's been through the committees and it's just about to hopefully be approved. So we're waiting, but it certainly has been recommended and it's on that path to approval, and we're expecting that that will happen. However, despite the fact that this test is great, there are still a lot of challenges. So there's the delayed access to testing. So in Australia, especially, children in rural areas are often not referred to clinical genetic services till quite late, and the average age of diagnosis is around 4 to 7 years. So if you can imagine, a child might present to their paediatrician at 12 months, but then the GP and the GP will go, well, let's see how your child's doing in 18 months. And then they go, okay, we'll send you off to a paediatrician, but then there's a 12-month waiting list. And so the paediatrician does some tests, and then no, we'll send you off to the geneticist. And so this waiting list is a problem, especially remote and rural areas of Australia. But even in Sydney, there can be waiting lists of up to 12 months to see a geneticist. So the other problem with this test is that despite the fact we can now test all the 1,500 genes, it takes the laboratory a long time to interpret one of these samples. And what helps them is what we call accurate diagnostic or clinical data. So if we saw a child and went, we think your child has a PAX1, and we said that to the lab, they could start looking at that area first. And so having this clinical information is really helpful for the lab. But as we start having paediatricians and GPs order the test, they won't be able to provide this clinical information readily. And so the question is, what can we do about that? And the question that I've mainly been interested in is these 60 to 70% of patients that remain undiagnosed following screening. So, yes. So, yeah.
Original.
So this boy was actually presented at a conference. So in Australia, we have a meeting 4 times a year called a clinical dysmorphology meeting, where we actually physically put up photos of our undiagnosed patients and put them up and say, does anybody have a patient who looks like this, or does anybody know what this condition is? Which, as you can see, it's a bit hit and miss. And, I don't get a chance to go to the London dysmorphology meeting or the American dysmorphology. I can't travel around the world to all these meetings. But in one particular meeting, there was this boy, and then in the audience, there was another doctor who said, look, actually, I do have a patient who looks the same. And so because they had both had their exome done but were negative, they then took these 2 patients and they went back down and looked at the exome, and what they were able to find is that they have a change in the PAX1 gene. And so this paper in 2012 really now said, well, PAX1 is a new intellectual disability gene, and that was found by this approach of matching the phenotypes. Then what happened is other doctors went, yes, I've got a patient who looks like that. And this gene then became part of the exome. So, we had so many genes we were testing, then PAX1 was added. So once PAX1 gets added, all these other children crop up around the world. And we then start to get a better feeling for the phenotypic spectrum. So we know that there are children with PAX1 who are less severely affected than these 2 boys, although these 2 boys were the first boys. And on their own, certainly I've seen lots of children now with PAX1 who on their own you wouldn't sort of think, well, they've definitely got some sort of, they don't necessarily look like they have a syndrome, if you know what I mean, but it's only when you put them together that you can see the similarity in these boys. So, but this is like looking for a needle in a haystack, trying to match up these children around the world who clearly have something. You know, a lot of people are doing a genome first approach where they're trying to match on gene variants, and that's a good way as well. But I think that what we need is an approach that combines matching genome variants with actually matching the phenotype, and that's where the power will come. So I was watching, I think I was watching television and read the conversation, and I saw some literature about Brian Lovell, who was a professor of IT at Queensland University. And Brian has spent 20 years of his life looking at the holy grail of understanding computer vision for CCTV. So that's not computer vision for a passport photo, that's computer vision for moving people who are blurry. And I wondered whether his technology could help us solve this problem. And so I met Brian and we did a pilot study, and one of the conditions, so when these two boys are in the database, Amber, of 5,000 images, and you take one out and you put the boy back in, these match as a #1 match here. And we did that with...
I can't believe that your talk involves PAX1.
Yes, there you go.
This is phenomenal.
Well, I think,
it's unbelievable.
When we did our pilot study, we only did 10 conditions, but one of the conditions we did was PAX1. So,
wow. Yep, keep going.
That's okay. So we did this pilot study which showed that this technology, this facial recognition technology, was good at matching children who had a known diagnosis. And so we wondered, could it be used for novel gene discovery? But we also thought, could it be used to make a diagnosis early, especially for inherited conditions where there's a risk? Parents often can go on to have a second affected child before their first child's diagnosed, so especially the X-linked conditions. Can we diagnose them early using this so that they can get access to testing earlier? Can we use this to help the labs prioritise which genes to look at, but also gene discovery? And this is just another example where this patient is in there, but she matches with this girl and she's got Cornelia de Lange. So they're different children, but you can see they have the same condition. So we decided that where we needed to go next was to establish an internationally scalable web-based portal. So the difference between this portal and Face2Gene is that Face2Gene isn't recruiting through parents, it recruits through doctors. But I can't show you the photo because I haven't got permission to show you all those images. But what we noticed when we did the pilot study is we had a girl who had a condition called Coulon-de-Vries, and we had multiple photos of her. And her baby photo matched with 4 other children with Coulon-de-Vries at the same age within the top 10. And then her toddler photo matched with another 4, and then her 10-year-old photo matched with another 4. And what it showed that this particular condition changes throughout life. You know how normally a baby's face changes, that even within these conditions, the face progresses and changes with age. So if you try to make an average face, it doesn't actually always pick up a young person's diagnosis, or it may not pick up the severity or the milder condition. So this system is, rather than developing a system to make an average or canonical face or to teach the software to recognize a condition, it's simply taking a face and putting it in the database, and as if it was doing facial recognition, it looks for the most common similar faces. So within milliseconds, it'll change through all the 5,000 photos and it'll say, this is the most common, this is the second most common, all the way down. And so then that actually works well because it takes in, this age problem. And then the algorithm looks at, well, how often does it match with somebody with another child with PAX1 or another child with Coulon-de-Vries within the top 10? Is it matching with children at different age groups within the same condition? So the algorithms are different to Face2Gene. And Face2Gene's good, it's just as geneticists, we haven't found it terribly helpful for conditions that we haven't been able to pick ourselves. And we think that this will start picking up more subtle children with more subtle features, because it's artificial intelligence. If I look at 10 photos, I can't remember what the 10th photo looks like by the time I get there, where this can remember it all, and it keeps it all there and can rapidly test that. So we realised we needed to launch a platform that was secure, and I really wanted it to have parent involvement. So I wanted parents to have somewhere to go when they had a child that was undiagnosed, that they could go to and try and sort of have this virtual meeting around the world where all these parents could have their faces matched, trying to look for new genes. So we set up this international portal. Now, because we're dealing with, when we were dealing with undiagnosed patients, the ethics committee obviously said we can't be giving results back to parents, and so now we have a system with where if a parent has an undiagnosed child, they have to nominate a doctor, and that doctor works with us to try and find matches or to work through various research projects. So the idea is that we're recruiting families who have children who are undiagnosed. However, we are also interested in recruiting families of diagnosed children, because we're doing lots of sub-studies to look at, well, how good is the algorithm, how can we improve the algorithm, and will the algorithm diagnose children at a young age? So the main differences to Face2Gene are it has parents and doctors involved. It allows parents to upload the photos. Because parents upload photos, they can upload a photo of a child at 6 months, 12 months, all the way through. And many of my families, I have like 15 photos from the one child, and that's incredibly powerful when you're trying to develop this sort of system. So this is our data, this is what it looks like, and we launched in 2019, and it's international. Because parents are consenting online, you don't need to have different ethics community ethics committees approved, because it's actually going out to the parents. So the front page talks about the project and gives, you can't see it here, but if you have a look at the project, there's lots more information about the project. We also have on the front page a Facebook page and a parent chat page. So if a parent comes in and they say, I've got a child with PAX1, and then another parent comes in and says, I've got a child with PAX1, you can actually find those other links, and so you can go back and just talk to people who have children with the same condition. The main reason we did that, because we know that most of these conditions will have their own support group, but it was for families that are non-English speaking families. The Arabic community requested this because Facebook will translate it into English with them, but they're having trouble finding support groups for their really rare conditions. They're so rare they don't even have a support group. So it's not trying to take over other support groups, but it's there if people want to use that. So who can participate? Individuals with syndromic intellectual disability who remain undiagnosed, or individuals who have a confirmed genetic diagnosis. So there's no reason, Amber, for other parents to contribute to this database, except the fact that it might help other parents. And I can talk to you about that, but that's a challenge, trying to get out to other parents who are really busy, saying, would you help us develop this database? We're also doing some...
We're more than happy to help everyone.
Thank you.
So we'll send you photos, it's not a problem.
Thank you. But what we're doing is, like, we're going out, we'll be going to the PAX1 international and national communities and asking them if they'll participate in a sub-project where we're trying to determine whether the system would have made a diagnosis by 12 months of age. So, and again, not necessarily that will help remote communities like, or countries like America, where it's really difficult to get exome sequencing unless somebody gives you a diagnosis, and also third world countries where they don't have access to a geneticist. So they're the patients. So if you're a parent, you go to the site, and I think my daughter's about to come upstairs. Excuse me for a minute. Grace, I'm just on the phone doing a presentation, so I might need you to be quiet. Thank you. She's lovely. Just got home from work. So the parents can go onto the site, they register as a parent, and then they add, it says add child, and they go in and they do all the consent online, and they can upload photos from their iPhone. We ask for frontal photos and a side photo, and face and hands are optional. And then we ask them, if they already have a diagnosis, you don't need to nominate a doctor if you are able to supply the genetic result. So if not, then we ask for a nominated doctor, and then the nominated doctor comes in and fills all that. So, but if you're a parent who doesn't have a diagnosis, you definitely have to have a nominated doctor, because we need the doctor involved in that diagnostic process. So the doctor receives an invitation once the parent's gone in. So let's just say, Amber, you went in and you thought, look, I will invite my doctor, they can provide the information. What was your child's name, sorry?
Lachlan.
Then your doctor would need to accept the invitation and complete that, and then submit it to FaceMatch. And what we found is only about 30% of doctors are accepting this invitation. So we're still looking at why that's the case. What we think the problem could be, there could be a firewall problem in some hospitals around the world. This is international. Certainly doctors are very busy, and my colleagues have said, look, you know, I have 80 unread emails in my box every day, and things go down to the bottom. So we're just trying to work that out. But that's another reason we've given parents the option who have a diagnosed child. You don't need to involve a doctor. You're just providing images to help, that they don't need to nominate a doctor. But when the doctor comes on, they complete all that. And the face matching results for the undiagnosed go back to the doctor, and then they contact the parents. But we provide a newsletter to parents once a year. And then the Facebook page. So the children who have a diagnosis, we're asking them to participate to see if we can improve the algorithm, test the algorithm, and look to see if it's going to be a good algorithm that can predict the diagnosis, help the labs, but help other countries that don't have access to testing. So this is the doctor form, which isn't really relevant here, but the doctor comes on and they fill out their bit. They just email the parent. So a doctor would come on. So for me, when I'm in clinic, if I'm doing a clinical exam, I would say to them, would you mind if I send you information about FaceMatch? If they say yes, I just go on here, put in the name, put in the child's name, put in the parent email address, all goes off to them, and then surprisingly enough, they do it, which is great. Parents are very motivated. They're more motivated than the doctors. I think, look, saying that, look, I know what the doctors are like. I mean, you know, we've sort of got 70 hours worth of work on our plate at any time, and so it always just feels completely overwhelming. But the parents go on once they get the invitation. It's an invitation from FaceMatch, so it says, you know, Dr. Dudding has invited John to participate in the FaceMatch project. Please read here about more information. They can read about it, go on, consent, upload all the images. That comes back to me. I just add some more extra information and off it goes. So there's this dual mode where the doctors can invite a parent, and we're hoping the doctors will start inviting undiagnosed parents, because I think it's getting to the stage that they need to have something to do with these children. That, you know, rather than, otherwise they just go, well, come back and see me in 3 to 5 years. And what we're saying is, rather than keep your photos, you know, keep photos on the computer, have them here in this secure database that can match with other children's photos around the world. And and that's our team. So Brian Lovell is computer vision. That is definitely a younger photo of me I need to change. Carlos is our bioinformatician. We call him the wizard because he is like just really mathematical, and so he understands all the algorithms and helps me with that. And we've got Jackie Boyle, who's a genetic counsellor. Jackie's also got a master's in bioethics, so she's the person who helps me with all our ethical dilemmas and getting through ethics. And I must say, this project was co-designed with community involvement, but also the ethics group. So we worked very closely with our ethics committee because we knew it was going to be problematic to get this through. And I think what's really interesting, Heather, is we've now got a community engagement officer on board the team. And so she's been very helpful. Like, she's rewritten all the text on the front page, and she's really helping with promotion. Yeah, and she's doing that on a philanthropic basis. So she's a mother of a child with a rare disease who's also a marketer and brander. So while she's home with her child who's unwell, she helps here. So we're hoping that we get more funding so we can pay her. So, but she's been great. So that's really all I had to say. If there's any questions, I'm happy to answer them. But I suppose for me, our next challenge, the first challenge was showing that, doing the pilot study, showing that it worked. The next challenge was getting some money, which we managed to get, to develop this very complex website, and to have a website that's available to parents and doctors all around the world. And Tess, although we say doctors, some of the genetic counsellors do this on behalf of the doctors. And so, the doctors, it might be something that genetic counsellors take on board. And part of our, so that's the second challenge, and that's up and going. And so I suppose the third challenge, which will be difficult, and I think we'll get an idea over the next 2 years how this goes, is that, Rome wasn't developed in a day, and this will take a long time to move this to something that is going to really make a difference. So what we're doing now is we're working with trying to engage with community groups who have taken the top 100 most common conditions, and that actually includes PAX1. And I think it might include FOXP1 as well. I'll have to have a look. I'm pretty sure it does.
Okay.
Sorry?
Yeah. So I think we've got about 8 families now, so I can plug it with those families.
Yeah. So, but we'll be actively engaging with family groups around the world. So our Facebook, we have ethical consent to communicate via Facebook with families, and so we're doing Facebook promotion through for the project, but starting with the top 100 most common conditions. So I feel quite confident that eventually parents will become engaged and they will contribute photos, because they can see. I'm hoping that they'll remember what their journey was like and see that that's something that will help other families. But I'm also aware that these families are incredibly busy because they have a child who has a disability, and so that's, we're asking a lot of them as well. So that's what we're doing. So part of it is just getting the message out to people, getting champions within each group. So Amber, I don't know if you're in contact with any of the international families, but looking for a champion. Yeah, so you might turn out to be a good person, but when we're looking for a champion and we've got all our marketing material, which really is, when I say marketing, it's more promotional material, but it's things that you can put on your Facebook page and stories about FaceMatch that you can share with that community. So once we've got all that together, we can come to you and say, are you happy to share this with the different organisations? Because I think for most of these conditions, there'll be an American foundation and a UK foundation, and then different countries will have their own parent groups, and it's about sort of promoting that to all these groups. And then the other thing we'll do is probably try and promote it to doctors who are actively involved in research in these conditions. So I think Han Brunner he'll be someone we'll talk to about the PAX1 families and things. So that's really it. Tess, have you got any questions? Probably that's a lot of the genetics you already knew, but it doesn't help to go over it. It doesn't hurt to go over it again.
No, I think it was great, and especially given I mean, there's genetic testing available and there is technology there, but funding is a huge issue. And if you can sort of narrow it down so you know more of what you're looking for, then I think that can be really helpful.
Yeah. Well, I think what was interesting is I had a colleague, and she saw a child out in outreach recently, and she said to the child's parents, I will do this exome, the trio exome, and if it's negative, you can go into FaceMatch. And they said, we want to go into FaceMatch now. And so, which was great. And so they did come into FaceMatch. And the child's face matched with the condition. And I said to my colleague, I said, oh, I think this child has this. And she said, oh, no, I've never seen anyone with that before. So she hadn't recognised that particular condition. So there's so many different conditions, and not all the geneticists are on top of them all, especially some of the newer ones. So the other interesting match that we've had, and I'm still waiting on the results, but little Charlie at the beginning. So Charlie's photo matched with a condition called her baby photo matched, and her teenage photo matched with children with a particular condition called Alpine syndrome. So I thought, wow, that's really interesting. We went back, and the gene for Alpine wasn't on her exomes that hadn't been tested. So we got quite excited and went back and tested that gene, but it was negative. So then we got a bit disappointed because we thought maybe we'd been able to work out what was going on. But her features were so typical that we sent her photos with consent to Dr. Zhao and Dr. Klein. And they said, we agree with you that she's got this condition. And we've got a number of patients who've got variants in the non-coding DNA that are affecting how that gene's functioning. And so they're actually now doing research on her, which wouldn't have happened unless we had highlighted that her phenotype or her face or her phenotypic information did actually match with that condition. Because so I think once we get a few things happen like that, then the doctors will come on board. Certainly my colleague was on board after I was able to say, I think this is what your condition, your child has, and she went, yeah, it does look like that. So it's just going to take a bit of time to get some focus and runs on the ladder, I think. But thank you for your time.